Hello, everybody. Today we're going to begin a brand new unit on cell communication. So cell signaling, as we will often call it, is going to be the main focus. So we're going to be talking about how cells can communicate with one another. And single-celled organisms can communicate with one another, but also multi-celled things can communicate with the other cells in the organism, and that's going to be advantageous. So these mechanisms for communication have evolved over time because that communication is necessary for the survival of that organism. So it says here, for multicellular organisms, communication between nearby or distant cells is critical for the survival of the species. Every single organism, even single-celled organisms like bacteria, have an advantage when they communicate or talk with one another. All right, so in what ways can cells communicate with one another? Well, we have a preview here showing different types of cell communication. So we have what's called endocrine communication. We have paracrine communication, we have autocrine communication, and juxtacrine communication. So very briefly, endocrine is long distance communication that goes through a blood vessel. Paracrine is short distance communication, which is between nearby cells. Autocrine, auto means self, so this is when a cell communicates or signals itself. And then juxtacrine is a physical interaction physically between two cells that will stimulate one another. All right, so what are signals and how do cells respond to signals? Well, a signal can be a physical stimulus. For example, light reaching your eyes, that's a physical stimulus. Or cell-to-cell signal, like when your heart cells beat, they are able to signal one another. Signals can also be chemical, and that usually refers to a hormone or a protein or some other chemical that is going to interact with some other part on a cell. Not all cells will respond to a signal. So just because a signal goes out doesn't mean that every single cell responds to it. Just like you don't pay close attention to every sound. As you study, you only pay attention to the important ones. To respond to a signal, a cell must have a specific receptor that can detect it. So if we are getting a chemical or a physical signal, the cell that should respond needs a receptor for that signal. One model, the signal transduction pathway, which is the main model we're going to talk about in this chapter, is the series of steps that lead to a cell's response to a signal. So here is autocrine signaling. So autocrine signaling, like we mentioned before, is when a signal binds to receptors that are on the very cell that secretes them. So here we have a cell in the middle, and this cell is secreting a signal. So notice we've got a vesicle here that's undergoing exocytosis, and it is spilling out a signal. And that signal is going to target two things. It is going to target itself. So why would it do that? Why would it signal itself? Because it may need what happens here. So the signal reception area. This may need to start a pathway that's going to cause some response. So for example, perhaps an immune response. So what could happen is if this cell has been infected by a virus, it might release a signal. It is then going to tell itself to start some sort of protective measure. And you can also see paracrine signaling here. It is signaling nearby cells, but notice what's happening. This cell does not have the green receptor necessary, so it is not going to undergo any change in response to that signal. So paracrine signaling, as we mentioned, they bind to receptors on nearby cells. Endocrine is when signals are circulated through the circulatory system and they bind to receptors on very distant cells. Often these are going to be hormone signals, and only very particular cells will have the particular receptor. And our point here, cells without receptors for the particular signal do not respond. So here we see again, local signaling. We have paracrine. So we have a cell producing a signal, and it is going to various target cells. Here we have a more specific example. We have a synapse. So these two cells are neurons. Those are nerve cells, and one nerve cell has been stimulated. So an electric impulse is traveling down this neuron, and that electric impulse is going to secrete a very particular type of signal called a neurotransmitter. That neurotransmitter is going to travel a very short distance to the next neuron, and it is going to signal the next neuron. So that is paracrine signaling, and this is referred to as a synapse. We're going to talk about the nervous system more at the end of the year. Then we have endocrine signaling. This makes use of our endocrine system, which you may have heard of. This is when one particular type of cell called an endocrine cell releases hormones into the blood, and those hormones travel all over your body, and certain cells might be target cells, although most cells probably will not respond to the signal. Certain ones will, and they may be very far away. So an example of this, your endocrine system may release a signal in your brain and it may respond to it near your abdomen. So it could be the pancreas responding, or it could be ovaries or testes responding to that endocrine signal. Juxtacrine signaling. So this is a physical interaction. The cells are touching, and we can see, similar to tight junctions and gap junctions, we see that there are usually junctions holding these cells together so that communication can occur. So cells can communicate this way. They kind of know when they're next to another cell, and they know when they aren't. These cell signaling pathways vary in their details, but a very common mechanism for them is allosteric regulation. So you already know what this term means. Allosteric regulation means that there is a spot on a protein that is going to change the three-dimensional shape of that protein if it is activated. So allosteric regulation involves alteration in the 3D shape of a protein as a result of the binding of another molecule. So that is called, what is that called again? That is called a conformational change caused by a molecule. So here we have some regulation. We've got a regulatory molecule that is causing a particular shape in a protein, and these three molecules can fit in. But when we have a slightly different regulatory molecule, it could change things to make it so that these things can't fit in. So we could activate a protein allosterically, or we could deactivate a protein allosterically. Hopefully, you're making some connections to our enzyme unit where we first learned about this. The entire pathway is referred to as a signal transduction pathway. So the entire signaling process, from the signals affecting a receptor, which is happening here, to the conveying of the message through the cytoplasm to the cell's final response, is called a signal transduction pathway. 3 parts: reception, so getting that signal; transduction, passing the signal on through the cytoplasm; and then response. Something is going to happen as a result of that signal. Here we see some examples of a reception. So notice we have an unactivated protein. It's in the membrane. And when a signal molecule comes along, notice what happens is that a conformational change has happened on the cytoplasm side of that receptor. And what this is going to do is that this protein can interact with another protein and activate it, right? So this protein, which would have been free floating in the cytoplasm, interacts with this one, and that causes a conformational change. And this protein can then interact with another protein and cause a conformational change. And this protein can interact with yet another one and cause a conformational change. And we can end up with a bunch of different responses. We might be making an enzyme, so we could activate an enzyme. We could activate a structural protein. We could activate a gene regulatory protein, which can help the nucleus make some other protein. It can help it transcribe. All right, so how do signal receptors initiate a cellular response? Well, receptors have very specific binding sites for their signals. Although any given cell is bombarded with many signals, it responds to just a few. So here we have the signal, which we are going to start calling a ligand, Ligand. I would underline this term. Notice that the ligand cannot fit into this receptor. This is the wrong receptor for the ligand, so it is not going to work. Over here, we have a cell that does have the right receptor, so the ligand can fit it. So this would be correct reception. And then we have transduction, and we will have some sort of response. A molecule that binds to a receptor site is called a ligand. That is the term we are going to use. Binding of the signaling ligand causes the receptor to change its 3D shape, conformational change, and that is going to initiate a cellular response, which we will talk more about later. The binding of a ligand to a receptor is like a knock on the door. And the binding is reversible. The ligand does not stay there forever. And if you think about that, that should make sense. We don't want the signal to last forever because conditions change, right? The signal is in response to something that's happening, some change that's happening in the environment or internally in the organism. So we only want the signal at the appropriate time. So this has to be reversible for it to work. Receptors can be classified by location. So some receptors are called cytoplasmic receptors, and some receptors are called plasma membrane receptors. Whether we have a cytoplasmic receptor or a plasma membrane receptor is really going to be determined by the type of ligand that we have. So here we have an example. Here is cytoplasmic on the left. So imagine there's a line going down here, and here's membrane on the right. What's the difference between these two types of receptors? What's the big difference? The big difference is on the left, this signal molecule is nonpolar, so it can make it through the membrane unaided. So here we have our inactive receptor. The nonpolar signal diffuses through the membrane on its own, and boom, notice the receptor is activated. So that is a cytoplasmic receptor because the receptor is in the cytoplasm. Here we have a polar signal, so this signal is not going to make it through the membrane on its own. So we've got to have a transmembrane receptor, and they have to go all the way through the membrane because you need part of it to be on the cytoplasm side. When the polar ligand fits into the receptor, it causes a conformational change on the cytoplasmic side, the interior of the cell, and that is going to cause something to happen. All right, and that is where we are going to stop for today. So if you have any questions, make sure you let me know.